Very nice. I will want to try and share my screen with you. It's kind of weird to hear myself speak. Uh, I guess nothing I can do about that. Yeah, so I hope you can all hear. Um, yeah, again, I want to really thank the organizer for giving me the opportunity uh, to talk about some of the work we've been doing recently in the lab. And also, thank you for the nice introduction. Um, so, we're going to talk about how blood signals control appetite and metabolism. Um, so, in my lab, we're sort of interested in how animals sense their environment and how they have these adaptive responses in order to coordinate metabolism and behavior and homeostasis. And so in order to adapt to different environments, animals need to be able to sense. Just a second. Uh, Can you still hear me? Let me check this.
I can hear you, but I think some people are saying that the audio is not
Maybe
great.
the problem with my computer. I'm just gonna try and go back uh, and share my window again here.
But actually, when you don't share the screen, I can hear you perfect.
What somebody said something it is. Sorry for this. Just
No worries.
turn this off. So animals need to be able to sense their environment and respond to it. So that involves different organs such as the gut and the brain and other tissues. And so we're interested in how organs communicate. And in order to sort of maintain an overall body-wide homeostasis, there are several important organs that need to communicate. So first, of course, food is taken up by the gut, and the gut needs to communicate level nutrients and so on to the brain, and nutrients can be stored in the fat tissue and muscle. So these organs communicate by several different mechanisms, and some of them involve hormonal signals. So we've been very interested in understanding the communication between organs. Now, the gut is a really important organ, and it produces several hormones that regulates a variety of processes. And one of the things that the gut does, which is really important, is that it controls metabolism, and it does so by producing hormones such as GLP-1 that affects the release of insulin, and that affects metabolism. But the gut also plays a very important role in controlling feeding decisions. So these gut hormones, such as GLP-1 and ghrelin, can affect appetite. So it plays a central role in controlling energy balance by both regulating the intake of energy as well as the expenditure of energy. Now, uh, we use in our lab the fruit fly as a model system and to study how hormones and how organs communicate. And we do that because it has these organ systems, including a gut and a nervous system, that perform very similar functions to mammalian organ system. It also has hormonal system, and many of these are quite conserved. And these systems here control metabolism and behavior. And flies have quite complex behaviors. Uh, so we really believe it's a very good system to try and understand how organs are communicating. Now, what we wanted to do, one of the things we wanted to do was to understand what are the signals coming from the gut and how is the gut processing information and how is it relaying that information to other organs to control behavior and metabolism. So we focused on the endocrine cells in the gut, and the gut is a very large endocrine tissue and has these enteroendocrine cells that produce sort of a cocktail of hormones. And we want to understand what the cocktail of hormones these cells produce and what are the things they regulate. So to do that, we did an RNAi screen. So we specifically knocked down secreted factors, transporters, and receptors in these cells in the adult gut of the fruit fly. So in my opinion, like doing genetic screens is really what flies are best for. But many times people just do a screen where they focus on one phenotype. And we wanted to do something a little bit different. So we wanted to do a multi-dimensional screen. Um, so we decided to look at when we knock these gut hormones down, how does it affect behavior? And we looked at both the feeding behavior and the sleep behavior and the activity of the animal. We also looked at how it affects sort of the physiology, how these gut hormones would affect metabolism and fecundity and survival under different types of stress. And then we did this in both males and females to understand what are the sex-specific differences in the gut and how it signals to tissues. And so when you do a screen like this, it's a lot of different parameters and screen about 250 genes. And if you put it into sort of a heat map, it looks something like this. And these, we just still taking the initial step to try and analyze the data, but I wanted to just show you a little bit about where we're trying to go with this. So up top here, we have all the phenotypes we measured, and out here we have the genes. So for each gene, we measured these many different phenotypes. We measured how the gene knockdown in the gut affected fecundity, but also how it affected resistance to stress. And we did both starvation stress, osmotic stress, and oxidative stress. We looked at whether the hormones would affect or the transporters would affect metabolism. We looked at how it affects the feeding behaviors and how it affected sleep and activity. And when you do these things, you can sort of like, you can start looking for correlations. And in this case, for example, the results are sorted for the animals or the genes that when we knock them down, give the biggest effect on starvation resistance. So up top here are genes that make the animal very resistant to starvation, so they live very long. And you can then start looking at correlations, and you can see that, for example, if you look at oxidative stress, there seems to be a lot of genes up here that are also marked in red, indicating that when animals becoming resistant to starvation, then they also become resistant to oxidative stress. And sort of down here on the bottom, you see the blue ones. And we're now doing a lot of different correlations and principal component analysis to try and understand sort of the phenotypic landscape of these things and the sex-specific differences. And I'm just going to go through a couple of examples here. So, for example, if you plot like here oxidative stress resistance versus starvation resistance, you see there's a quite nice correlation in that. And on top of that, if you put the amount of TAC, which is the amount of stored fat the animal has, you will see that animals that are up here are very resistant to starvation. They also resistant to oxidative stress, and they generally have a very high level of TAC, which makes sense because if you're resistant to starvation, it's very likely it's because you have a lot of energy stored, so you can survive a very long time if you starve. There are also things that do not correlate as well. So if you, instead of plotting oxidative stress, you plot osmotic stress, you don't really see a very nice correlation between osmotic stress resistance and starvation resistance. So we think this indicates that these are actually different. They're sort of different mechanisms and regulated differently. So in our data set, there's a lot of things that are correlated that I think are really exciting, like sleep is obviously correlated with feeding, and sleep is also correlated with fecundity and all these different things. So this is sort of where we're going with this now. But without talking too much of this, I want to talk about some of the most striking hits that we found on the screen and how that sort of led us to understand how the gut processes nutritional information and relate that information to other organs. So if you sort for starvation resistance, we observed that one of the genes that gave the biggest resistance to starvation was a gene encoding a hormone called aldostatin C, or just SC, and it's the fly version of a human hormone called somatostatin. So we want to understand how this hormone is affecting basically resistance to starvation. So first we did some follow-up experiments. Let's see if this is coming. So we did some follow-up experiments, and we looked at both males and females, and we could already see from our screen that it seemed to be working in a sex-specific manner, and we confirmed that. So if we specifically knock down this aldostatin C hormone in the gut, in the internecine cells, we saw no effect on the survival under starvation in males, and we confirmed this with RNAi, and we did tissue-specific CRISPR. However, In females, with both RNAi knockdown and CRISPR knockout, we saw a very strong increase in their survival after starvation. So we want to understand if that was linked to metabolism. So we looked at how this SC would affect the mobilization of energy, because animals can be very resistant to starvation if they have a very high level of stored energy, but it could also be that whatever stored energy they have, they would mobilize very slow. So first we looked in males and we didn't really see, and top here we looked at glycogen levels, and on bottom we looked at tag levels. And we looked in the fed state and in the starve state. And we didn't see any big effect in males of knocking down allostatin C in the gut. However, in females, it sort of seemed to be able to, when the animals were in the fed state, there was very little effect of knockdown. But when we starve the animals, control animals seemed to mobilize and deplete their energy, but the flies lacking SC in the gut did not deplete the energy as fast. So that seems to tell us that gut SC worked in a sex-specific manner to promote the mobilization of energy in starvation in females. So we wanted to make sure, because there's a lot of gut hormones, and many of these hormones are also expressed in the brain, including allostatin C. So we wanted to make sure that we actually only knocking down allostatin C in the gut and not in the brain. So we did a number of experiments to prove that. Here's just a couple of them where we can show that with this driver we're using, and we specifically reduce SC transcripts in the gut and not in the nervous system. And we confirmed this with staining, showing that we couldn't find any detectable differences in the stain of allostatin C in the brain, but we specifically depleted the SC stain in the gut. So the phenotypes and the energy mobilization phenotypes that we saw, we sort of attributed that to the gut-derived SC. So the next thing we want to understand what is causing the release of allostatin C from the gut. We knew that it seems to be working during the starvation state to promote the mobilization of energy. So we wanted to understand, is SC then released from the gut when the animal is starving? And what we found was that if you go from fed state over here, and here are interneurokyne cells in the gut, to the starved state, we could almost not detect allostatin C in the starved state in the midgut. And here over here you have the quantification. So there is less SC peptide, but when we looked at the transcript levels of SC, we didn't see any change. So that indicates to us that they produce an amount of allostatin C, but they release the SC when the animal starves. We took another tool into use. We specifically first took the so-called allostatin C driver, and we drove expression of DAP, and we focused on this region of the midgut called the R3, which is the middle midgut. And with that tool, we drove a calcium reporter. So it's a calcium lexase system that reports the activity of the cells. So what we found with that tool was that the calcium activity of the allostatin-producing cells in the gut was increased in starvation condition. And we had a higher number of activated cells. So altogether, that indicated to us that when the animal is starving, the SC cells in the gut become activated and they release SC, consistent with the idea that SC is working when the animal is starving. We are then wondering how these SC cells sensing the lack of nutrients, and we looked into TOR, which is a main nutrient sensor, and we asked whether maybe TOR would regulate SC, and to do that, we blocked the activity of TOR specifically in the SC-producing cells, and we looked at whether that affects SC release. And what we found was that an inhibition of TOR led to lower SC levels in the gut, indicating that inhibition of TOR, which is what would happen during starvation, is causing release of SC. So we think that at least TOR is involved in the process that leads to release of SC from the gut in animals when they're starved. Now, we also measured expression. When we knocked TOR down, and actually expression of SC went up. So the lower level of TOR of SC combined with the high expression indicate that it is being released. Now, we want to understand how can gut SC mobilize energy when the animal is starving. And there are two key hormones that control metabolism in animals. It is insulin and glucagon. And insulin is sort of released when the animal is after a meal and is telling the tissues to take up and store energy, while glucagon is released when the animal is fasting or starving and is telling the tissues to mobilize energy to keep blood sugar levels normal. So these two systems maintain a sugar homeostasis in the blood. So we had sort of looked, we sort of by now knew that during starvation, SC is produced by the gut, and we want to see whether SC is regulating metabolism by acting on the fly glucagon, which is called AKH, or just adipokinetic hormone, which is produced in these cells you see here in green. Now, first we asked, is the elatostatin C receptor produced in the cells that make AKH so SC actually can work on those cells? And we found that SC receptor 2 is specifically expressed in these cells. You see here this co-localization with AKH. And the next thing we wanted to know was, does SC receptor knockdown in the AKH producing cells give a phenotype and increase starvation resistance? And sure enough, we confirmed that if we knock the elatostatin receptor down in the cells making AKH, the animal will become resistant to starvation, just like if we knock SC down in the gut. And this phenotype is similar to animals lacking AKH. So the next thing we looked at was whether SC receptor knockdown in the AKH producing cells affect mobilization of energy. And sure enough, we found that animals, if you knock the receptor down in the cells making AKH, they mobilize their TAC slower and also their glycogen, similar to when we knock SC down in the gut. And this phenotype resembles the loss of AKH. So basically, loss of AKH, which is a glucagon version in flies, also leads to slower mobilization of energy during starvation. So the SC knockdown in the cells making AKH phenocopies the loss of AKH itself or the loss of SC in the gut. So it was very likely that SC from the gut would signal to these cells. Now, we wanted to make sure that the gut SC through the SC receptor, it's actually regulating the release of AKH. So when we looked at AKH levels, we found that AKH levels are reduced in animals when they're starving. However, if we knock the receptor down in cells making AKH, these animals do not release AKH when the animal is starving. And we confirmed that by looking at transcript levels that that basically in control animals, the expression of AKH is a bit up, but still they have less AKH over here, indicating releasing it, and that effect goes away with knockdown of the receptor. But the more important experiment was do we see the same thing if we knock SC down in the gut? And that's exactly what we do. If we knock SC down in the gut, now again the animal fails to release AKH when it's starved. So it tells us that gut-derived SC is basically promoting AKH secretion when the animal is starving. Now, the role of AKH, just like glucagon, is to sort of maintain metabolic homeostasis and presumably to release stored energy to maintain blood sugar level normal during fasting. So we asked whether SC is actually required to maintain sort of like blood sugar level normally during starvation. So if we do that, we measure the glucose levels in hemoglobin, and sure enough, we found that if animals that lack acid signaling in the cells making AKH, they cannot maintain normal glucose levels during starvation. So they basically become hyperglycemic. So this really shows that animals they need to release ST from the gut to promote the secretion of this glucagon-like hormone to prevent hypoglycemia under starvation. Now, we also want to understand a little bit, dig into whether this actually this shows that ST from the gut affects metabolism, but it's also been known that this AKH hormone is sort of affecting feeding. So we asked whether gut SC also affects food intake. And we found that if you knock SC down in the gut, the animal eats less over short time periods. This is a short-term food assay. If we knock down the receptor of SC in the cells making AKH, they eat less. And if we knock down AKH itself, they eat less. So this tells us that gut-derived FC promotes food intake really, and the mobilization of energy. We confirmed this in another long-term essay, and we found the exact same thing. Now, uh, so based on that, we proposed the following model for how during nutritional stress, when there's no food intake, how SE is coordinating metabolism of food intake. So the enterocrine cells in the gut release SE, and when the animal is starving, and that at least involves four, we think. And SE works on the cells and make AKH, and promote the release of AKH. And AKH promotes the mobilization of energy that is stored in the fat tissue or the adipocytes. And at the same time, AKH probably work, we think, on the brain, and it's something we're looking into to control feeding. So it promotes sort of hunger and food intake. Now, so that's what happened. We found this hormone that works when the animals starve, and this shows a gut hormone that controls metabolic decisions by working on the fly glucagon. Now, we really want to understand these gut hormones that control appetite, and because they're really key to maintaining energy intake. And I want to just give you a little bit of perspective on the importance of these hormones. Now, I come from a small country, and the biggest, the most profitable country in Denmark is called Novo Nordisk. And the reason why this company makes so much more money than even the number two company is because they sell, the patients use the treatment based on GLP-1 therapy. And GLP-1 is a gut hormone. And the reason why more and more patients are using this is because more and more people become obese. And it turns out that GLP-1 treatment is really good for weight loss. And the reason for that is that GLP-1 suppresses appetite. So gut hormones that suppress appetite are really maybe the future, I think, of obesity treatment, and they're very, very important. But so we started looking into appetite, but it turns out that appetite is not just appetite. Appetite can be specific. So animals can have appetite for the specific nutrients they lack, and that is a mechanism that sort of ensures that the animal gets the nutrition it needs. So animals living off like a diet that's high in carbohydrates and fat can, as sort of an unbalanced diet here, can be hungry for protein food, while animals that live on a carbohydrate and fat-rich diet can develop appetite for a protein-rich diet can develop appetite for carbohydrates and fats. So very little is known about the mechanism that control specific appetite, and especially the hormones that control specific appetites are not very well known. So we wanted to go back and use our screen to understand what are the gut hormones and what are the mechanisms from the gut that affect appetite. And we sorted the gut screens here for genes that regulate appetite or food intake, basically. And we found a hormone called neuropeptide F, which is the fly version of a mammalian hormone called neuropeptide Y or NPY. And actually, we think this one is also homolog of a hormone, of a related hormone called PYY. If we knock down NPF in the gut, the animals basically overeat in our screen. And then what we did in the screen was we used this flick system that monitors like feeding behavior. It actually doesn't monitor consumption of food, but just the interaction with food. And when we knocked down NPF, we found that the animal eats, and this system only measures sugar feeding. So the animals basically have increased sugar feeding behavior. So indicating that somehow this gut NPF is important for regulating sugar feeding behavior. So we wanted to next measure actual consumption of food. So we used a short-term dye assay, and we found that if we knock NPF down in the gut, the animal will overeat in a short-term dye assay. And if you do a longer-term dye assay with only sugar food, knocking down gut NPF leads to a strong increase in the consumption of sugar. So this was done with a driver that sort of targets all the enterocrine cells in the gut, and we wanted to have a more specific tool. So we used a driver to knock down NPF in the gut that's based actually on the NPF promoter enhancer. So we used what's called the NPF L4, and that expresses GAL4 in both the brain and in the gut because NPF, like SC, is expressed in both tissues. But we combine this with another tool called R57C10 to suppress the neuronal activity. So with this driver, we can knock things down only in the NPF positive cells in the gut. And we confirm with this other tool and several other RNA lines that if you knock NPF down in the gut, the animal will overeat in a short-term assay, it will overeat sugar in a longer-term assay, and it will have increased sugar feeding behavior. So uh, all these results suggested that NPF is a gut hormone that inhibits sugar consumption. So we wanted to see, does NPF actually regulate the preference for dietary sugar? So we gave the animal the choice between 1% sugar or 10% sugar. And what we found was that if you knock NPF down in the gut, that you actually get, it increases the animal's uh, preference for high concentration of sugar. And we confirmed this again with the other driver as well. So we did it in several different ways. So this really suggests that NPF from the gut inhibits the preference for high concentration of dietary sugar. Now, so while we found that the NPF knockdown leads to increased consumption of sugar, we found a very interesting thing when we presented the animal with only the opportunity to eat protein food, protein-rich food. In our case, that is yeast. That's the main source of protein, so they give the animal only yeast. Now, animals with knockdown of NPF eats much less than control. So while they overeat sugar, they eat much less protein food. So that indicates that somehow that while the NPF suppresses sugar appetite, it actually promotes the intake of protein food. And we started thinking a little bit about sort of the biological context and meaning of that. So we have used for all experiments mated females. And in flies, when the female goes from virgin to being mated, uh, that's associated with an increased preference for protein-rich yeast food because the females that are mated need more protein to reproduce and produce eggs, basically. And this has been shown, this preference for yeast has been shown by elegant work by Carlos Rivera's group and others, that it's regulated by the actual mating process. And during the mating process, the transfer of a hormone called sex peptide from the male to the female is responsible for this. And then another interesting thing is it was also known that the sex peptide in mated females is actually also sort of potentiating the release of NPF from the midgut. So we asked whether NPF was sort of important to suppress the sugar appetite in mated females and lead them to eat more protein-rich food to reproduce. So we looked into both virgins and mated, and we found that in virgin females, there was basically very little effect, if any, of knocking down NPF in the gut. However, in mated females, there was a very big effect. And if you look here on the gray, which is the control, you can see that going from virgin to the mated state, females increase their consumption of yeast. But if the animals that lack NPF, they don't increase their consumption of yeast after mating, indicating that it's required to do this. We also used another system called the fly pack that was developed also by Carlos Rivera's lab, where you can give the animal also the choice of eating either 10% yeast or 10% sugar. And we found that in virgin females, the NPF knockdown in the gut had almost no effect on the preference, but in control females, you see in gray, they really have strongly increased their preference for yeast, and this doesn't happen without in animals that lack NPF in the gut. So this really indicates that gut NPF regulates the food choice and promotes the consumption of protein-rich food after mating. Now, we want to understand how are the cells in the gut basically sensing sugar and how they're releasing NPF. And we identified in our screen a sugar transporter called SUT2, which is similar to mammalian includes 7. And the interesting thing about SOT2 was that when you knock SOT2 down in the NPF producing cells in the gut, the animal would overeat sugar, just like the knockdown on NPF itself. However, we gave them the option of only eating yeast. They would eat less yeast, so completely the same phenotype as NPF knockdown. We also started looking into the preference, and in this case, we used a two-choice diet, giving the animal the choice to eat sugar or protein food. And when we did the preference assay, and it might be a little difficult to see here, but in control animals with gray here, in virgin females have lower preference for yeast than the mated females over here. But in animals that lack NPF, SO2 in the NPF cells in the gut, they don't increase their preference for yeast in response to mating. Basically have the same. So this indicates that the loss of SO2 in the NPF cells phenocopy is the loss of NPF itself, and it's important to switch the appetite after mating. Now, that opened the possibility that SO2 is somehow involved in the sugar sensing process in the NPF cells and is regulated in the release of NPF. So we looked into that. And again, we used the calcium XA tool to look at the activity of the NPF cells in the gut. We also measured NPF itself, and we labeled all the cells making NPF with TD tomato. And what you can see is you just feed an animal. As our time goes after feeding the animal with sugar, the NPF cells up here in green are becoming activated. At the same time, the NPF cells starts to be depleted for NPF, indicating that they are actually releasing NPF. Now, we wanted to see if SO2 is required for the release of NPF during this process. And what we found was that we knocked SO2 down in the NPF producing cells in the gut, NPF accumulates in these cells, indicating that it's not being released. So these data suggest that the sugar transporter 2 is sort of involved in the sugar glucose-stimulated NPF secretion in the gut. So we think that's part of the mechanism that controls NPF. Now, so we know that NPF is when the animal goes from a starved state to a state where it feeds sugar, that causes a release of NPF. So we want to understand, we are characterized as C, we want to understand how is NPF regulating food choice. And we asked again whether NPF may act on these AKH producing cells and make this glucagon-like hormone. So the next thing we did was we knocked down the receptor for NPF in the cells that make AKH. Okay, so and then we did the same assay where we gave the animal a choice of eating either 1% or 10% sugar. And we found that the knockdown of NPF receptor in the cells making AKH increases the preference for high sugar concentration. We confirmed this with 2 different assays. And we next wanted to see, is the NPF receptor, can we detect the expression of there? Is it really there? And sure enough, we found the receptor is expressed in the cells making AKH. And we also found, when we looked at actually AKH levels, that if we knock down NPF receptor, the AKH levels are lower, indicating that they're releasing NPF. So this suggests that NPF is actually inhibiting the release of AKH, while I showed early on that SC is promoting the release during starvation. So this suggests that during sugar feeding, NPF inhibits release of AKH. Now, we also want to look at whether the knockdown of the NPF receptor affects the preference for sugar and yeast, just like NPF knockdown in the gut does. And again, we found no effect in the virgin stage of knocking down the NPF receptor in cells making AKH. But while control females increase their preference for yeast after mating, we didn't see any increase if you knock down NPF receptor. So again, this shows that both the gut NPF as well as the NPF receptor in the AKH producing cells are required to increase the preference for yeast after mating. Now, we wanted to show that the effect we were looking at from NPF receptor in the cells making AKH were actually mediated by AKH and not some other hormone. So to do that, first we knocked down the NPF receptor, and we found that increases the sugar intake. compared to the control over here. But if we knock down NPF receptor over here and at the same time knock down AKH, we completely eliminate that effect. So that tells you that AKH are mediating the effect of NPF and NPFR in cells making AKH. So if NPF is inhibiting the release of AKH, we would expect the opposite phenotype from knocking down NPF to knocking down NPF receptor or NPF itself. And that's exactly what we found. So we found that knocking down NPF receptor and the cells making AKH increase sugar intake. Knocking down AKH makes complete opposite. They decrease sugar intake. Now, that's also consistent with a lot of previous literature showing AK or indicating that AKH is a hunger hormone because the loss of AKH leads to less intake of food. But many of these studies had looked at only sugar food or mostly sugar food. But however, if you look at yeast, so protein food, now we found the complete opposite. NPF receptor knockdown leads animals to eat less yeast food. AKH knockdown animals to just overeat yeast food. So that indicates that AKH is not just a hunger hormone, and it suggests that NPF inhibits the preference for sugar and promotes the intake of protein through it, through suppression of AKH. And AKH is really a hormone that regulates food choice. So we're very interested in finding that. So we started looking into what is AKH actually regulating the preference between yeast and sugar? And we said if NPF is inhibiting AKH to promote the preference for yeast, if we just inhibit AKH by knocking it down or deleting it, we should promote the preference for yeast. And that's exactly what we found. We found that knockdown of AKH with RNAi or AKH mutants, they have strong increase in preference for dietary yeast over sugar. We also thought that in at least the way we thought about it was that our model said that when females go from virgins to being mated, that causes a release of NPF from the gut in response to sugar. And the role of NPF is to inhibit AKH to switch the appetite towards intake of protein food. So then the high signaling of AKH in the virgins would be what is promoting sort of like the appetite for sugar. So we looked into how just deleting AKH would affect sugar protein intake in virgins. And we found that if you delete AKH, the virgins just overeat protein food. And while control animals, they increase their protein intake after mating. You can see here in gray, there is no difference in AKH mutants. They just have increased intake of protein food. We also did the preference using the FlyPad system again. And again, confirmed that if you have an AKH mutant, they have strong increase in preference and these are virgins. Even in the virgin state, they have strong increase for protein food. Now we wanted to see what happens if we, instead of knocking down an AKH, but if we activate AKH signal, can we then switch the system in the opposite direction? So now we use this TRP1A tool, which allows us to activate the cells making AKH. And again, in control females, we see that if you go from virgins over here to mated, it increased preference for yeast over sugar. However, if we activate AKH release, even in mated females here, they have very strong preference for sugar. So really supporting that AKH can suppress the yeast preference. And if we knock down AKH, it's a complete opposite phenotype. So we did another experiment, which is also sort of like an interesting one, at least when you start looking into this, is we also video recorded the animals in an arena where they had either access to sugar food or protein food. And over here you have control animals, and you just sort of visualize. I have to go just to visualize for you the difference. So here there are sort of like 3 yeast patches, and there are some invisible sugar patches. And what you can see is that the control animals, they sort of like explore both patches, whereas the AKH mutants over on the right is basically sitting on the patches of yeast, and they almost do not go to the sugar patches. So it's a very striking difference in the preference for yeast versus sugar. And you can quantify this, that the position on the food, and it really shows that AKH is regulating this food choice. Now, we sort of sum this up in the following model to explain at least how this NPF system is required after mating to drive consumption of more protein. So as the animal is mating, the female is being mated, the transfer of sex peptide from the male is inducing the midgut NPF system. And midgut NPF does a couple of things to suppress AKH signaling. I didn't have time to go into all of them today, but I show you that once you go from starvation conditions to sugar intake, via SUT2 in the NPF cells, it causes a release of NPF. NPF acts directly on the cells that make AKH to suppress AKH release. What I didn't show you is that NPF actually also acts on the cells that make SC to prevent the release of SC, because SC is promoting AKH, so it sort of shuts down SC signaling. And then NPF actually works directly on the adipose tissue, the fat tissue, to counteract AKH signaling and to prevent the mobilization of energy. So it seems that NPF is really strongly shutting down the whole AKH axis. And by doing this, NPF is suppressing AKH, and that switches the appetite. So by suppressing AKH, you get less appetite for sugar and more intake of protein. So I've been trying to sum these findings up in a sort of a little bit of a different model and put them into how we think about them now. So the first part I showed you was that during starvation, you have release of SC from the gut, or the fly version of somatostatin, and this promotes the release of AKH from the cells that make AKH, and AKH promotes the intake of the appetite for sugar, while it inhibits the appetite for protein. So when you've been starving, you're most likely looking for an easy energy source, which is sugar. And at the same time, AKH is promoting the release of sugar into the blood to maintain blood sugar stable. Now, when the animal's then eating sugar, the gut is releasing NPF, which is a fly version of a human hormone called also PYY, which is produced by the human gut. And NPF inhibits AKH, so AKH goes down. And that means basically that the animal stops releasing the stored energy because it already has consumed sugar, so you don't need to sort of mobilize any stored sugar. And at the same time, after a sugar meal, this leads to an appetite for protein-rich food. Now, we think this system quite elegantly shows how the gut is really sort of controlling both metabolism and feeding decisions and to maintain homeostasis. And I want to just give you a short insight into what we're working on at the moment, because we're really interested in how the gut is processing nutrition information. So there are still other nutrients that we are trying to understand, and one of them is protein food. So what happens when the animal is eating protein? How is protein being sensed by the gut? And we have some data on it, and this is one of the things we're working on now. So we think that the intake of protein leads to the release of another hormone called tachykinin. And we have already found that if you have an animal and you look at the tachykinin positive enteroendocrine cells in the gut, now if you compare 10% sugar food to 10% protein food, the tachykinin producing cells are much more active on the protein food. At the same time, if you look at tachykinin levels, they have much less tachykinin. So it indicates that on protein food, tachykinin cells in the gut are now becoming activated, and they're releasing tachykinin. So we have evidence beyond this that shows that tachykinin is actually also working, at least in part, on AKH. We found the tachykinin receptor here is expressed in the cells that make AKH, and we have a lot of other evidence for this. So if this is true, we're trying to understand why why, if you eat protein, would you want to release AKH? And certainly our model right now is that it makes sense why both SC and NPF regulates AKH in opposite direction in response to starvation and sugar. But once an animal is eating protein food, in our model, and the data we have, it indicates that the kind is promoting release of the AKH, just like sort of starvation. And we think it makes sense because if you eat protein, the release of AKH would sort of promote appetite for sugar food because now you just had protein. But I think there's one other very important thing, and that is if the animal's eating protein, uh, but no sugar, protein food will also lead to release of, for example, insulin. And if you release insulin and you're only eating protein, it will mean that your blood sugar is going to drop because the cells will take out all the sugar. So in that scenario, you need to also release this AKH of glucagon to actually make sure that you release some sugar into the blood. So we think that explains, and that's why animals during protein feeding need to release this glucagon like AKH factor. So this is sort of what we're working on. One of the things we're working on now, trying to understand, and then we're trying to understand what are all the transporter and sensing mechanisms in these various groups of internal cells in the gut. There's many different cells that produce a cocktail of different hormones, and we are trying to dig into that. With that, I want to thank the people that funded the work, and particularly I want to thank the very talented people that did the work in my lab, and then I want to thank the people that provide the reagents, and I really want to thank you for the opportunity here to talk about our work. And if you have any questions, I'll be happy to try and see if I can answer some of them. And I'll try to stop sharing my screen.
Thank you so much. This was a fantastic talk, and thank you for sharing all your work. So I'm going to start with the questions from the audience. So if you guys now have any questions, you can place it here in the Ask a Question button or even in the chat that I will read it out loud for Kim. Well, actually, the first one, it's not even a question. It's just great talk, which I completely agree. So while we give some time for people to write down their questions, I want to ask you, so this is kind of like more maybe a big question. How is the relationship between the sex peptide and how the gut hormones change after mating? Can you speculate on that?
Yes, it's a very good question. I mean, we don't know how sex peptide is affecting NPF in the gut, basically. We see it does because we've done enough different things where uh, if we made females to males that lack sex peptide, we don't see the change in the gut. We can also put sex peptide receptor down in those neurons in the female that's supposed to receive the signal, and again, we don't see the effect. So we know it's working on the gut, but it's kind of mysterious how it's doing that. And of course, it's a very exciting thing that we would like to understand. We would also really like to understand how AKH is controlling choice. So we think AKH is working on the brain or some neurons to do that. So that's one of the things we also want to look into.
It's so interesting. Okay, so we have one question from Rita. Interesting talk. Maybe I missed it, but how do you envision this system working in males in which there is no mating
Yeah,
syndrome?
so that's also a great question. How would I? So the thing about it is that I'm trying to understand it. In our screen, I would say like 30 or 4% of the genes we knock down, we see they work in a specific way, maybe 40%. So many of them are sex specifically, and for elastase C and in PF, we sometimes see the opposite effect in males, and we simply have not had the time to look into how these hormones work in males, we either see no effect in the male in these cases, or sometimes the opposite effect. So my feeling about it is that there certainly are like a lot of things that are not sex specific, but there's also like more things that you probably imagine that are sex specific, and so we want to use the screen as like an entry point to try and understand the sex specific uh, effects in the gut and how the male and female gut are processing things differently and then nutritional inputs and other things. So that's what we want to try and do some analysis on the sex specific differences, because I think it's kind of really interesting, and I think that we're quite sure that the age really occupies a central role in sex specific regulation of behavior and metabolism. So that's quite clear.
Thank you. Another question. Could you estimate in total from your very cool functional screen how many different neuropeptides in total are required to regulate feeding behavior and food choice in
Yeah, I
Drosophila?
can give you the answer of how many we see in our essay regulates feeding behavior. And off the top of my head, I don't remember exactly because we screened the neuropeptides and screened also neuropeptide receptors because we think that the gut is actually, like we showed from NPF, NPF work on SC cells. So we think there's a lot of processing going on in the gut where the gut is processing the information and or different cells in the gut are communicating. And then, so in response to one input, they're producing a certain output. Uh, so I don't exactly know. We also have a lot of transporters, but I would imagine that we are talking about a dozen of neuropeptides. There's some effect, and yes, some effect, not strong effect, some effect. And then there are also hormones that are not neuropeptides that we find have strong healing effects that are more stress-related hormones. So yeah,
Another question, I wonder how gluconeogenesis from amino acids in an amino acid only diet will fit your model, especially its effects on TK.
can you say that again, maybe?
Yes, sorry. I wonder how gluconeogenesis from amino acid in an amino acid only diet will fit your model, especially its effects on TK.
Yeah, it's a good idea. Uh, right now, we think we think the way TK attack is being the release is being regulated and also lead the uptake of amino acids into the cytokinin producing cells because we can knock down the certain amino acid transporter in those cells, and then we get a phenotype that resembles the loss of cytokinin. So that's what we're working on right now, sort of like similar to STRIP2, but just for cytokinin cells and protein intake. Uh, that's a good question. And I don't know very much about it actually, but another thing I've been thinking about also in terms of amino acids is how we always think about how AK glucagon promotes release of stored sugar and fat to maintain blood sugar, but I think also AK is probably affecting the uptake of protein into cells. So it probably affects the hemoglobin protein levels. I don't know. That's what I'm speculating about.
Okay, another question. Do you think some similar mechanisms operates in mammals? Thanks.
Yeah, nice question, man. Yeah, so just as an example with this NPF, so the mammalian gut makes a so mammals have a family of NPY hormones, which are the mammalian version of NPF. So flies only do one NPF, and it seems that NPF is working in the brain and in the intestine. And in mammals, they have NPY, which is working in the brain, and then they have the PYY, which is produced by the gut. So we think that the gut on NPF occupies the role of PYY. And it's certainly known that PYY is a hormone that regulates satiety in mammals, and it's suppressing appetite. So at least that seems to be also the role of NPF in fly gut. But in fly, we just shown that it's specifically related sugar appetite while it promotes the appetite for protein. And I think basically most big drug companies have a program on PYY. So, but I talked to the people that are working on these things, and they say we don't know anything about drug hormones that control like food choice. So it'd be interesting to see if there some of these things are conserved. And I think some of the hormones seem to be doing conserved things. In humans, the human gut also makes somatostatin, for example, which is a version of aldostatin C and things. So I think some will be conserved, not all, but some.
Okay. I think I can hear you well, but I think from the audience, people cannot.
Uh,
Again, like your voice is a little bit mm, different now. I will try to leave my microphone on. Maybe that may solve the problem, but I know my computer is somehow an helicopter. So maybe if the sound doesn't improve, I will try to mute myself again.
I also have
So
a little echo on my computer. Don't know if that's a problem.
yeah, I don't know. I feel that it's a little bit chubby, just like
Okay.
if you speak slow, I at least I think maybe it will improve, but I can hear you. I think it's I don't know if it's because I'm just inside this, but I can understand you well. Okay, so another question. Well, it's a little bit long. So a more big picture question. Given that the same type of neuropeptides and hormones have many different types of effects on different tissues depending on the state of the animal, what do you think is the next step for the field to try to understand how these signals regulate homeostasis? Do you think we can continue looking piece by piece, or do we need a different type of approach, technology? Asked in a different way, where is the field going?
That's also a good question. And at least part of the reason we did a multidimensional screen was that we wanted to see if we could learn something from that we couldn't learn just looking at a single hormone and how that's affecting feeding or metabolism and and that's something we're still working on see what we can. I mean, we can see all these correlations between things, so we want to try and understand what that tells us and see if we can sort of define finite spaces and behaviors that are really and things that are correlated and related. So I think that's at least part of where we think we want to go with this is to try and understand if we can understand things in the context and not like a single hormone level. Uh, and then the other thing I would like to understand is how is the gut actually processing information and how is it like taking certain inputs and processing that and using certain outputs that goes to other organs. And I don't know if I answered the question of where it is going, but we would like to understand. There's so much uh, like when you look at things, you look at static conditions, but then you start exposing the animals to different types of stress or nutritional conditions. Maybe very important to see, and I think we need to learn how are these factors responding to that also over time. And one could imagine maybe that it's single cell sequencing or something like that. You can see how the cells that produce the peptides are responding over time to different types of conditions, and we have some ideas that we want to try and do that and sort of superimpose that on functional states.
Thank you. Another question, NPF GAL4 can drive the expression in both gut and CNS, but according to the single-cell RNA-seq database in adult, it is also expressed in other tissues. I don't know if that is more a comment than a question.
Yeah, I mean, I actually am not aware of those tissues that express NPF beyond brain and gut, but it's an interesting thing. And also, somebody said these peptides have many different functions, right? So one can also imagine you look at how it's gotten because obviously working on AKs, it's might also work on muscle. We show the work on fat, maybe it's even working on the tissues, and I think that's one thing that'd be nice to understand. In our case, we use two drivers. We use the gut NBF or the NBF4, which would target the gut and presumably these other tissues, but we also used an internal specific driver, or specific, but we use the driver called 4, which targets the internal cells in the gut along with small subset of neurons in the brain. So I think by using those tools, at least we can show the difference coming from the gut in this case.
Okay, and we have two more questions that I will try to ask also a little bit in a rush. I will leave mine for the Zoom meeting, and if anyone else have more questions, we can you can also join with us. So, do you have any idea how NPF receptor knockdown in insulin-producing cells will affect the food choice behavior, or do you expect any effects at all?
Good question. So yeah, these are the insulin producing cells. We looked at the AKH producing cells. We also knocked down NPF receptor in the insulin producing cells, and we didn't observe any obvious changes in food choice or feeding behavior that at least agreed as far as I remember with the phenotype we got. So we thought that it was that the phenotype, at least if any, could not be explained by a gut to or signaling to the IPs. So at least in our case, we excluded that gut is regulated through the IPs and then as far as we can. And yeah, and it brings me also to think about there's this whole, of course, gut brain signaling thing that I think is super interesting. But my feeling is also that a lot of the gut signals probably relate information to organs that are sort of anatomically, physically close, right? And the species just sits right on top of the gut. So it's very easy for gut hormones to reach the cells at an age. And I suspect that many of the gut hormones also communicate to cells, maybe not in the central brain, maybe in the ventral nerve because that's very close to the gut. So it might be very easy to relay information between those tissues. So tissues that are close, I think, might communicate more.
Okay, last question. Just wondering, where do you place the brain in your model? If I understand it correctly, it is the gut sensing the nutritional state and responding accordingly. Could you please comment on this and also correct me if I'm wrong? Thanks.
Yeah, great question. Maybe I wasn't clear enough about that in my presentation. So we've shown that the gut works on these cells that make this glucagon like AKH, and that regulates feeding decisions. We are now looking in our model, AKH is regulating feeding by acting on the brain, and the receptor for AKH is expressed in neurons in the brain. So, and AKH is only expressed in these cells, basically. So, right now we're looking into how AKH, by acting on the nervous system, would uh, control food choice and feeding behaviors. That's kind of our model. And yeah, um, we also think about so hormones such as tachykine are released from different like regions of the gut, like the anterior, middle mid gut, and posterior. And I think hormones, even the same hormone released from different regions in the gut can have different effects. And I think that's because the part of the gut that it's close to communicate more with the tissues it's close to, basically. So that goes for both tachykine and other things. And I think that's also part of the things we have to think about in the future is that how can the same hormone have different functions? And maybe it's related to the fact that it's actually communicating mostly with the tissue that is like the source tissue is close to the target tissue. There's a better communication.
Okay. So, yeah, thank you so much for the talk, for the answer to all these questions. Again, we will meet by Zoom now, and I'm going to place again the link here in the chat for those of you, because I think that it already went a little bit high. So yeah, thank you so much, Kim. Thank you everyone for joining us today. We will have next week Pierre-Marie Yedo, and he's going to talk about microbiota, cognition, perception, and memory. So I hope to see you all next week as well.
Thank you so much.
See you. I will also send you the link through email. I don't know if you can see it now in the chat.
Yeah.
You have it?
I have the link. I should be.
Okay, perfect.
So I
So
can disconnect here from the broadcast.
yes, I will stop now the event so we can move there. The recording will stop and everything will stop, but we will keep the discussion. See you, see you.